recording now. So we are now recording. Cool. So this is going to be our midterm review, and we're just basically going to go from beginning to end, but I'm not going to like go into too much detail about the questions. I'm just going to gloss over big picture ideas. All right. So this can be used as sort of like a supplement to your studies. A lot of my quizzes questions are going to be reflective of what you'll see on the midterm exam. So make sure that you go over the quizzes questions as well. Okay. All right. So before we begin, do you have any specific questions of anything that you would like clarification on, just right off the bat?
I mean, I think I already asked mine in class the other day about what specifics we need to know as far as, you know, antibiotics and antivirals and what type of cell they target and stuff like that. If we don't really need to know like specific
Yeah,
antibiotics that target
that's going to
specific.
be your next exam. That's going to be the final test.
Okay.
Because we haven't really talked about antibiotics too much yet. We're going to talk about antibiotics in chapter 5.
Okay.
So chapter 4 is like antimicrobial type. It's like disinfectants, sterilization procedures, things like that. That's chapter 4. Chapter 5 is going to be antibiotics.
Okay. Cool.
Okay. So don't worry about antibiotics yet.
I didn't mean necessarily antibiotics, but like drugs that are used or effective against like HIV/AIDS, or drugs that are effective
Right.
for Hep C, or vaccines that are available, like specifics like that.
We'll just get through it then with the exam review here. So if anything like that comes up, then we'll talk on those points. But for the most part, drugs are going to be dealt with in the final exam.
Cool.
All right. So with that being said, let's go ahead and get into it. In microbiology, I already gave you a quizzes question on this the other night, but in microbiology, you're basically studying things that you can't see with the naked eye, right? So you're looking at things. By the way, you can't see my screen, right? I'm not screen sharing. Okay, good, because that would really suck. You have like all the questions. Anyways. So in microbiology, you're studying things you can't see with the naked eye. So in other words, what are you going to be using to be able to visualize those things? We used it the other night. What was that you looked into when we did gram staining?
The light microscopy.
Yeah, light microscopy is a good example. That's going to be like the most commonly used tool to visualize, single cellular organisms, peripheral blood smears, even like histology for like cancers and things like that. You're using light microscopy. Also, electron microscopy, right, is going to be a very commonly used, well, not as common as light microscopy because it's like expensive equipment, but you can really see greater detail, right? So you're looking at things smaller than bacterial cells, right? Viruses are a good example. Even other components of the cell, like you can see mitochondria, you can see Golgi apparatus, you can see all the little, cellular, the little components within a eukaryotic cell, viruses, etc. You can also use confocal microscopes. We don't really talk too much about confocal microscopy in this class, but it's another example of something that you could use. I already asked you that question, but in the quizzes question, but it's good to bear repeating. Cool. So when you look, there's going to be a couple questions on the exam where you literally look at a cell, a eukaryotic cell, and you're going to have to identify what's what in the cell. Okay. So let's go over some of the big things that you might see in a eukaryotic cell. What's like what are the major things in a eukaryotic cell?
Nucleus.
Yeah, nucleus. Excellent. What else might you see as part of like the membrane? What's it called? Endomembrane system. It's part of like the nucleus, and it's also part of some other organelles that are associated with the nucleus. You can see other things in there too.
Capsules or capsids? No.
No.
Or like organelle bound.
Yeah, other organelles, so membrane-bound organelles. So you sometimes in the nucleus you might see rough endoplasmic reticulum, endoplasmic reticulum. Those are going to be different components. You might see Golgi apparatus. Golgi apparatus is what's going to allow for vesicles to be shuffled off to be excreted outside the cell. And you might also see, well, you'll see mitochondria, right? If you're looking at plants, you'll see chlorophyll and sometimes mitochondria. What do you see in the mitochondria?
Uh,
What does it look like?
like rough, is that where the rough ER is?
No, so mitochondria is going to be, it looks like it's kind of like a
Just
bean
little
shape.
beans,
Yeah, it's kind of like a bean shape, and
the labyrinths.
it's got an inner mitochondrial membrane, which is kind of like a labyrinth.
Yeah.
And that's where you actually see like the electron transport chain and all that. Just know what that organelle looks like. Know what the organelles look like in general, and just like kind of like the general function. Like Golgi apparatus helps to like repackage different components so that they can be released from the cell through exocytosis. The mitochondria, just know what it looks like. It's going to be associated with generation of energy, right? ATP production, that's going to be in the mitochondria. So just know your general organelles and what they look like if you see an image of that. Cool. So you said just a second ago, did you say spore a second ago? Or you said capsule, right? You said capsule. Yeah, so something that's kind of related to capsules would be something else. What happens when a cell is, if they're capable of doing this, what happens if they're subjected to like a lot of like stress? For instance, if it's a obligate anaerobe and they're exposed to oxygen, or if they're in like nutrient deficient environments, or if they're just like an inhospitable environment altogether. What do some of those bacteria do to protect themselves? To go into hibernation?
Sorry, I have to go on mute real quick. Sorry.
No, you're fine. Any idea at all, Jennifer?
I think that was one of the ones that I wrote down the other day.
Yeah, we talked about it the other day as well. So hint, Clostridium tetani, Clostridium botulinum, Clostridium difficile, as well as Bacillus anthracis, Bacillus cereus. Those are examples of these types of bacteria, and they can change they can like do something. They can form something specific.
Spore?
Yeah, spores. Those are spore-forming bacteria, right? And the spores are going to be made up of a lot of different things like calcium. You might see glycocalyx in there as well, but calcium is going to be one of the major components where it helps like protect the DNA from harsh environmental conditions, whether it's oxidative stress, if it's an anaerobe, right? Or if it's exposed to UV light. So UV light can actually damage DNA. So those spores can protect them from UV light. It can protect them from nutrient deficiency. So they can live in the environment for like over 100 years. So that's going to be spore-forming bacteria. Our spore is going to be reproductive.
No.
No, right. Spores are non-reproductive. Vegetative cells are going to be reproductive. Okay, so we didn't really go over this as much yet. It's the second part of chapter 3, but what is the genus that causes malaria? It's okay if you don't know. We didn't talk about it yet. We're going to talk about it in more detail on Tuesday. But Plasmodium, if you've heard of that before, Plasmodium is malaria. There's Plasmodium falciparum, which is the #1 cause of malaria. There's Plasmodium ovale. There's Plasmodium vivax. There's actually Plasmodium malariae, which, interestingly enough, is not the #1 cause of malaria. Plasmodium falciparum is. What does malaria fall under? Is it a fungus? Is it a bacteria? Is it a virus? Is it a protozoan? Is it algae? What is it?
Protozoan?
Protozoan.
Yeah, good. It's a protozoan or a protist. That's great. So malaria, in terms of protozoan, because we did talk about this early on in the semester, we're going to talk about more detail on Tuesday, but malaria is like the #1 protozoan in terms of like historical impact on humans. Malaria has killed more people than any other infectious agent throughout human history. right? More people have died from malaria. So that's kind of a interesting side note. So Plasmodium, like a lot of people don't really focus on Plasmodium, but Plasmodium is a significant category of microbes, right? Hey, what's up? How you doing, Danielle? So there's Plasmodium. Other ones that fall under protozoans are going to be like Giardia, right? Entamoeba histolytica is another example. We're going to talk about this one as well towards the end of the semester, but Negrelli fowleri, which is the brain-eating amoeba. That's a pretty nasty one. Trypanosoma, there's Trypanosoma cruzi, brucei. Those are all different ones. We're going to go into those later on, but Plasmodium, I think, is, they're some of the most significant infections, especially malaria. Very, very significant. A lot of people still get affected by that in those malaria endemic regions. But know that malaria is going to be falling under that like protozoan category. Make sure you know innate versus adaptive immune system. That's going to be pretty heavily tested here. So in terms of innate immunity, what's going to be like, you remember when we talked about innate immunity, we had like different lines of defense. We had the like primary lines of defense.
Mm-hmm.
You remember that? When you hear innate immunity and you hear primary line of defense, think barriers. Okay. What's a really good example of a barrier?
Skin.
Yeah, skin, mucosal membranes, those kinds of things. Those are going to be like innate, like the barriers that are associated with the first line of defense, innate immunity. Okay. So that's one thing. We'll go into more of that afterwards as well, but let's see. Pretty straightforward on that one. That's straightforward as well. Let's see. I'm skipping over the ones that I find like ridiculously easy that you guys don't need further clarification on. All right. I'm trying to figure out how to go over this question without giving too much away. All right, so let's talk about two things. Let's talk about capsule versus biofilm. Okay. So what is going to be one of the most like significant components of biofilms?
Like what? Like
what's it made out of?
Glycoprotein, glucose,
polysaccharides. Yeah, so
polysaccharides.
like sugars, like very sticky sugars. My question is, polysaccharides is going to be the major component, but also glycocalyx can also be a component. Now, what else do you find that contains glycocalyx?
I feel like I just wrote about that one.
Another place where you find glycocalyx and polysaccharide are going to be in capsules. Okay, so you can find them in both. But one thing to keep in mind about biofilms is that you see a lot of polysaccharides, but you also do see glycocalyx as well. Okay, so you do see both within a biofilm. Okay, does that make sense?
Mm-hmm.
Okay, awesome. That one's too easy to go over. Let's see what else. All right, what do you find in viruses? What are the major components? There's two things that you're always going to find in a virus, and then sometimes you might find a third one as well. Don't look at your notes. Try to like see if you can remember it off the top of your head.
Nucleic acid.
Yes. Which is going to be found within what? What surrounds the nucleic acids?
Nucleus or nucleoid?
No, that's bacteria.
Oh, shoot.
Nucleus is eukaryotes. Nucleoid is bacteria or prokaryotes.
So nucleic acid.
Uh-huh.
What is
What surrounds the nucleic acid? It protects it.
Capsid?
Capsids, which are made up of what?
Is it like the envelope structure that you were talking about?
No,
or-
that's not the envelope. The capsid is different. The capsid is made up of
Phospholipid layer?
no, that would be the envelope.
Yeah, I'm stuck with them.
Okay, so you have nucleic acids. Let's draw it. All right. So I'm gonna share a blank screen. So you guys see my screen here?
Yeah.
Okay, cool. So here's all your nucleic acids. My little crappy drawing. Okay, then you have your nucleic acids, which are gonna be found in your capsids. What are your capsids made of?
Protein?
Yeah, there you go. Cool. So you're always going to have nucleic acids, right? I'm just going to write Na. And then you're going to have proteins, which make up the capsids, right? This together is called a nucleocapsid. Nucleocapsid. Sorry, I'm using my mouse, so my handwriting sucks. And then sometimes you do see that like little phospholipid bilayer on the outside. Okay. So then you do sometimes see lipids, right? So sometimes you see lipids, all right? And then you have like your spike proteins and all that stuff, okay? Cool. I'm not going to draw the whole thing out here. This is my Rembrandt of the night. I'm not going to go into too much more detail. So nucleic acids always, whether it's DNA or RNA, never both, right? Proteins, which make up your capsids, combine, those turn into the nucleocapsid, and then lipids you might see sometimes, only if it's an enveloped virus. All right. Rock and roll. So I'm going to stop sharing. All right. No more sharing screen, right? You don't see my screen anymore?
Right.
Okay, good. Let's move on then.
Just you.
Just me. Oh, how boring. All right. Let's see. What kind of structures do organisms use to move? Let's specifically think about like protozoans.
Like the flagella.
That's one. What's another? Cilia. Yeah. So paramecium, for instance, are going to be surrounded by a bunch of little hair-like projections, cilia, so they can like swim around the water. There's also a third way that protozoans can move. What's that?
Cilia, flagella, and
Think about amoebas. How do
pseudopods.
Pseudopods, yeah, exactly. So pseudopods work. They extend a little projection, they latch on, and then they drag themselves over. They extend, they drag. So that's the pseudopodia. So it's like a fake foot, and that's like considered like amoeboid type motility.
Is that like what your little favorite, that little thing is? You have pseudopods?
My favorite is Giardia. Giardia moves with flagella.
No, that little guy that looks like a little living creature.
Oh, the paramecium. Those are cilia.
No, it's got like a weird name. It's like the Targod or something.
You had it on your
Oh, tardigrade.
Yes, tardigrade.
No, tardigrades are, I think they fall under animal, to be honest.
They look like they have like literal like feet with little claws on it.
Yeah, but they don't fall under any of those organisms. They're eukaryotes.
Okay.
Sorry, so are protists, but they're like multicellular. So I think they fall under Animalia, if I remember correctly.
They look like little animals, like little bear things.
Micro animals, because they're multicellular. They're not just one single cell. Yeah, anyways, that's not tardigrade. Tardigrade is going to be different, but pseudopodia, if you look up amoeboid movement, so for instance, Entamoeba histolytica does that. Negrelli fowleri also does that. They'll like shoot out these little projections and they'll drag themselves to move. Tardigrades literally move with their little legs. So they actually like they ambulate like an actual animal. So it's kind of cool. All right, moving on. That's so
Are
fun.
we going to have to label like the different bacteria, like the shapes and
Different shapes of the bacteria. You should have a general understanding of all the different shapes of the bacteria. You should have a general understanding of like what they're named when they're round, if they're round in chains, which would be called what?
Streptococci.
Yeah, exactly.
Yeah.
And if they're round and they're all bundled up in a group, in a bunch.
The cocci only staph can form with cocci bacteria, right? With those,
Yeah, so if it's like bundled up like a bundle of grapes, that'll be Staphylococcus.
right? So they'll never be like staph bacillus. They'll never be staph vibrio or staph. It'll always only be staphylococcus.
As far as I understand, Staphylococcus is only associated, like if you see staph as a prefix, that's only going to be associated with cocci. I don't think there is a Staphylobacillus. I don't think that's a thing.
Okay.
There is a streptobacillus. There's a lot of them, as a matter of fact. Bacillus anthracis is a streptobacillus. Lactobacillus, like if you go to like Sprouts or whatever and you buy some like probiotics, those are going to be mostly lactobacillus. Those are streptobacilli. You'll see them hanging out in chains if you look under a microscope. But I don't think there is a streptobacillus. As a matter of fact, you sparked my curiosity here. Is there
Staphylobacillus.
Staphylobacillus? There is a Staphylobacillus, and it falls under lactobacilli group. But mostly for lactobacillus, oftentimes it's going to be streptobacillus. But I guess there is a Staphylobacillus, just meaning clusters of rod-shaped bacteria. As far as I know, there's none that really cause like disease. Well, let's see if there is. That was a good question.
It just seems like that's all you ever see is just.
Yeah, because we mostly focus on ones that cause disease. And just on a preliminary research, or sorry, Google search, I'm not really seeing anything that actually causes disease in Staphylobacillus. Like when we talk about microbiology, especially in the realm of like, pre-nursing and pre-med, or even in like med school and stuff, we're mostly dealing with stuff that cause disease, right? And from what I understand, at least on like a, very superficial search, I'm not seeing anything that causes disease.
Okay.
If you see anything, let me know. I'd be interested because I think that's kind of interesting. Cool. Let's see. What else is on here that we need to know? Okay. I'm skimming over the ones that are way too easy. That's gonna be a waste of time to go over. Okay, even though most of you guys, did well on the gram staining procedure, let's start from, the beginning to end. What are the 4 steps in gram staining?
Crystal violet, right? Is that what it's called?
Yeah, and what's crystal violet? What is that considered?
Good question.
It's just considered a primary stain.
Primary stain.
Yeah, so primary stain. Then after you add the primary stain, crystal violet, what's the second thing that you add?
Iodine.
Iodine, good. And that's a mordant, right? So it allows the crystal violet to adhere to the inside of the gram positives. Then you do a third step.
Alcohol.
Yeah, so you do a decolorization step with alcohol.
And
then what's the 4th and final step?
Secondary stain, the safranin.
Counter stain.
Okay.
Counter stain.
Is it gonna ask counter stain or safranin? Like, is there any like?
It's good to know which one. Like, it's good to know both. Know that generally speaking, the primary stain is going to be crystal violet. The counter stain is generally going to be safranin. Okay. So that's pretty straightforward. Let's see. Oh, here's one thing that you guys should know. Okay, so oftentimes bacteria are going to be named after the things that they cause. Like, let's see. Let me try to think of one off the top of my head. Okay, anyway, scratch that. Sometimes bacteria can be named after diseases, but they're also going to be named after just like their features. So, for instance, there's like a Micrococcus luteus. that is it looks like a staphylococcus, but it's yellow. So it forms yellow colonies, and luteus in Latin means yellow. There's like Micrococcus rosiae, and that's going to be causing like kind of a rosy pinkish color type colony. And then you can name them after where they're found too. So for instance, the bacteria that we worked with the other night, Staphylococcus epidermidis or epidermidis, potatoes, that's gonna be found on your epithelial layer, also found in your mucosal membranes too, in like sputum. Staph aureus, I know. Oh, she left.
Oh, she said she was having computer issues. She was going to try
Yeah.
again.
So si tú hablas español, what's oro?
Gold.
Gold, excellent. Or AU is on the periodic table, right? So if you see Staphylococcus aureus, what does that mean? It's a golden Staphylococcus, okay? So that's one example of how bacteria are named. So
Okay,
if
so
you have a general understanding of like how some of these are named, you might be able to, it will help you kind of like better understand how to answer some of these questions.
is it gonna ask like, I mean, I know you maybe can't specifically, do we need to know like what is the bacteria that's typically found on the skin that presents itself in yellow gold color colonies, or
Yeah,
do
sure.
we just need to know?
It's good to know that. And there's the two bacteria that in terms of what they look like and where they're found, that's one of them, what they look like, or what the term is derived from. One thing that you do with Staph aureus, if you grow them on a mannitol salt agar, we don't do that in this lab, but we do that at my other lab. When you grow them on a mannitol salt agar, it actually changed the color of the agar. So it goes from being a pinkish color to kind of like a yellowish golden color. That's like kind of a good way to kind of remember that Staph aureus turns gold when you grow them on a agar, like a mannitol salt agar.
Oh, I did that. Yeah.
For Staph epidermidis, know that you find it both on your skin and you find it in your sputum. Okay. That's one thing to keep in mind. Okay, so moving on. So there's coliform bacteria as well. So coliform refers to colon, right? So GI, coliform. There's a lot of bacteria that's going to be found in your GI, like members of the Enterobacteriaceae are going to be found in your GI tract. What's a really, really common bacteria that's found in your GI tract? What's the number one cause of UTI, urinary tract infection?
I
Yeast.
don't think it's E. coli.
Huh?
No.
Oh, okay.
Yeah.
So
That's
we're
such
gonna
an
get
easy question. I don't know the answer.
Hi, that's okay. Okay, so E. coli. Escherichia coli is.
That's what I said.
Yeah,
Oh,
I
you
thought
said
you
that.
said
Okay,
no to
good.
Jen. I was like, wait, what?
I didn't hear you. Sorry. So E. coli is going to be one of the most common bacteria you find in your GI tract. It's mostly found in your large intestines. That's why when you're potty training young girls, you tell them wipe front to back, right? Because you want to avoid doing any sort of like cross contamination. Because E. coli is also the number one cause of UTI. Okay. So that's something to keep in mind. So that's going to be like coliform bacteria, coliform, think colon. Okay. So E. coli, cholera is another good example of that. Cholera is something that would be affecting the large intestines or the colon. Cool. So let's keep moving on here. We already talked about this one. Okay, so in terms of growth for bacteria, what are the four major things that bacterial growth is dependent on? There's like four major things.
pH, temperature, light. No. Oh, it's like pH, temperature is salt.
pH, temperature, oxygen,
Oh yeah, anaerobic and
and then water. Water is going to be a big thing as well. Salt can definitely affect bacteria. Like, for instance, there's different types of augers, like mannitol salt augers, for example. They're going to select for those gram positives. Gram positives tend to like saline, salty environments. Gram negatives don't really like salt very much. But in terms of those 4 different components, pH, temperature, oxygen, water. Now, a lot of the bacteria that affect humans, right, pathological organisms or pathogenic organisms, they're going to like the type of environments that our body provide, right? So temperature, a lot of them are going to love body temperature, right? So 37 degrees Celsius, 98 degrees Fahrenheit-ish. They're going to also like the pH levels of your body. What is going to be your pH level, for instance, in your blood? What's the average pH? You don't have to know the range, but just like, what's the average?
No, I'm not even gonna say anything because I sounded stupid enough tonight.
No, that's okay. That's what you're here for. You're here to learn. And I sound stupid all the time. So,
It's like 7 something.
yeah, 7.4. 7.4 is like average pH, right? And so bacteria are going to love, the ones that affect us the most are going to love to like exist within that pH level, right? So if they're going to affect us, they're usually going to want to be in that, that type of pH. So around 7.4-ish. Okay. All right. Moving on. All right, let's talk a little bit about the adaptive immune system. So the adaptive immune system is subdivided into 2 different major categories. What are they?
Humoral and cellular immunity.
Awesome. Let's focus on humoral. What does that mean?
Isn't it the antibodies produced by the B cells?
Yeah, which B cells turn into what?
The T
Plasma.
cells? No, I'm sorry, plasmids.
Yeah, exactly. They turn into plasma cells. And then, so that's when they differentiate and proliferate, and they secrete immunoglobulins to target specific antigens. And then what about the cell-mediated immune system? What exactly is the cell-mediated immune system targeting? Or sorry, what cells are involved?
The T cells.
Yeah, T cells. Great. So T helper cells, and then cytotoxic T cells as well. Natural killer T cells. So if you know that B cells and T cells are the major cell types that are involved in the adaptive immune system altogether. What can those cells do once they get exposed to an antigen? They mount the immunological response. What happens after that once they get exposed to an antigen? What's the big thing about the adaptive immune system? You're adapting to something. You're adapting to things, right? You're adapting to foreign invaders, foreign antigens, things like that. So if you're adapting to those things, what exactly is happening with those cell types?
Like the memory cells.
Yeah, exactly. That's exactly the thing. They form memory cells. So you can have memory B cells, you can have memory T cells. We're mostly going to be talking about memory B cells, but there's memory involved, right? So when you adapt to something, you're going to actually remember once you get exposed to that antigen again, then when you have a second exposure, that reaction is going to happen much more easily, much more rapidly, versus like the primary response is usually about like 7 to 14 days. Secondary response is like 5 days, around there. So it's really rapid, and it's going to be a little bit more aggressive, right? Because you already have memory to that. So that's one of the key components of the adaptive immune system. Okay. Okay, so let's switch gears a little bit on bacterial components. So let's talk about gram positives. I know we're beating a dead horse with this, but what's the major component of a gram positive?
Teichoic acid, thick peptidoglycan.
Peptidoglycan, yeah, and teichoic acid for sure. Teichoic acid allows for that vertical scaffolding, which allows for horizontal scaffolding of those NAG/NAM subunits. Cool. And then what about gram negatives? What's the major component of gram negative?
LPS.
which is what?
Lipopolysaccharide.
Awesome. That's great.
And
Fantastic.
then the thin cell wall. And it's
Yeah,
like
so they have
either
a much,
red or pink.
if it's a gram negative, it does have peptidoglycan, but it's gonna be really thin in comparison to a
Mhm.
gram positive. Exactly. All right. All right, let's talk about the Kirby-Bauer test a little bit. Tell me what the Kirby-Bauer test is. What does it show?
Antibiotic resistance.
Yeah, exactly. And if you do see antibiotic resistance, okay, if it's susceptible, what do you see?
The zone of inhibition.
Yeah, exactly. So you put the little tab on the plate, you grow like a bunch of bacteria on that plate. You have the tab, and then around the tab, you're going to see a clearing, and that's called the zone of inhibition. That means that the bacteria is indeed susceptible and sensitive to that antibiotic or antimicrobial.
So that's how they measure susceptibility, is by how many, what is it, like nanometers or
You
millimeters?
could do it by distance, but that's not a really, it's not a truly accurate way of doing that. The best way to like really determine what works best, they're going to be doing like the minimum inhibitory concentrations as well as minimum bactericidal concentrations. You could do that, but the zone of inhibition is going to be a pretty strong indicator that the bacteria is susceptible to the antibiotic or antimicrobial.
Do we need to know about the minimum dose, like which one is when you start to see a decrease and which one is when it's gone? Like, do we need to know?
You're not going to necessarily be testing on that. It's good to know the concept. So let's just kind of conceptualize it real quick. So you have like maybe 10 different vials, right? 10 different vials of bacteria, but then you have 10 different doses of an antibiotic going from a small dose to a higher dose, right? So maybe say in your 4th or 5th vial, let's say you have 4 milligrams, just for the sake of simplicity. You have 4 milligrams in that vial. You see the bacteria, it's no longer growing. It's still there. It's not dead, but it's no longer growing. That's the MIC. That's the minimum inhibitory concentration. At 4 milligrams or of whatever compound that is, that's when the bacteria doesn't grow anymore. It's still there, but it's not going to grow. Then you up the dose a little bit, right? It's still inhibiting the growth, but then you get to maybe vial number 7 milligrams, and they die, right? That's going to be the minimum bactericidal concentration. So now you know, cool, if I want to stop the growth, 4 milligrams. If I want to kill the bacteria, it's going to be 7 milligrams. So conceptually, that's kind of the way to think about it, right? So minimum inhibitory concentration inhibits growth, minimum bactericidal concentration kills it. Make sense?
Yeah.
Cool. I think Bonnie froze. All right.
I think she did too.
I think so too.
Well, my son just got home, so he's probably got on his computer to have the same problem Claire was having with these.
You had a really blank stare. I'm like, am I being confusing? I was like, wait, she's not moving.
I usually, if I don't get something, I usually won't just stare at you. I'll try.
That's hilarious. I love it. All right. Okay, we talked about that. We talked about that. Oh, actually, okay. So we're going to talk more about this when we get into, let's see, what chapter is that? Did we already talk about this? Okay, so what's the difference between active immunity, passive immunity, and what are the subcategories of each of those? Artificial versus natural, artificial versus natural. Let's start with passive immunity. What does passive immunity mean?
One, like an infection that's transferred in active immune components from one person to another.
Yeah, so passive immunity means that you're transmitting immunoglobulins. The immunoglobulins are already made, already formed. You're just like transferring that to a person, right?
To clarify that, and I'm only asking because there's a questions on the worksheet we have this week. For it to be passive, does it have to be like fall under direct, like where it's person to person, like it has to be an actual one person giving it to another person? Or can it be indirect contact as well?
Active immunity could be derived from indirect contact. So think about this. Think about you're walking, you open a door, right?
Yeah.
Maybe there's like a virus on that door. Take, for instance, coronavirus. Okay. Simple example. You grab the door, you like touch your eyeball or something, and now you just introduce that the virus into your system. You get sick, right? That's indirect contact. You touch the door handle, right? So that's indirect contact. But now your body gets sick. Your T cells, your B cells all get activated. Your B cells now are going to start developing antibodies against that virus, right? So that's going to be considered active, not artificial, but Jesus, what? Natural. That's going to be considered active natural immunity.
Okay.
So that's your body is developing the antibodies. If you had active passive immunity, that would be an injection, right? A vaccine that gave you components of the virus, right? Say maybe, for instance, like influenza virus, right? So if you take an influenza vaccine, you're going to be getting components of the virus. So you're pretending that you're basically tricking your body into thinking that's actually getting sick. And then that's also going to be active immunity, but it's going to be artificial, right? Because you're getting.
You said active passive. Did you mean artificial passive?
Okay, wait. Are you talking about for what the example I just gave you?
Yeah, you said active passive is going to be, and then you started.
Totally convinced. I'm totally confusing myself. So let me do a white screen.
So artificial passive is when you get a vaccine and then your body responds to. So like, for instance, it would be the getting a mild case of influenza after receiving the flu mist vaccine would be.
Okay, let me just do it this way. Okay, so you have active on one side and passive on the other side. Active can be active natural and active artificial. Passive can be passive natural and then passive artificial. Okay, so active immunity means that your B cells, T cell, lymphocytes are all getting activated. Mostly your B cells, because they're the ones that are actually producing the immunoglobulins, right? So I'll just follow Y's here. Those are your like little immunoglobulins. So that's going to be active immunity, where you're actually getting your body is producing the immunoglobulins. That can happen either naturally or artificially. Okay, naturally, active natural would be you get sick, right? You get exposed to the virus and all of a sudden you're sick. Okay. So let's think like let's think about the flu, right? So influenza virus. I don't know. I'm just gonna write it as I. Influenza virus, you get sick. All of a sudden you get exposed to, I don't know, influenza A or whatever. And now your immune system is going to develop those immunoglobulins as a reaction, right? So now your plasma cells are able to develop those specific immunoglobulins against that specific type of influenza. So that's natural. Artificial would be you give the person an influenza vaccination. Okay, so you give that person a vaccine. That's a really crappy syringe, right? But you give the person a vaccine, right? That's artificial. It's exogenous, right? But what you're doing with the vaccine is that now you are stimulating those lymphocytes, the B cell lymphocytes, right? Now those B cell lymphocytes are going to start producing immunoglobulins as well. Okay.
So are they kind of like the opposite? Like one is exposed and then you get the immunoglobulins, where the other one is the immunoglobulins are given and then basically your body accesses.
No, don't get confused with that yet. Okay, we're not talking about exogenous immunoglobulins. Passive is going to be strictly involving immunoglobulins.
Okay.
That's it. Okay, immunoglobulins that have already been produced, already been formed. Okay, with active immunity, you are resorting you are relying upon your B cell lymphocytes to get exposed to the to get activated and proliferate so they can develop the immunoglobulins for you. You're resorting to your own B cell lymphocytes. Okay, that's active immunity. Whether it's natural when you got sick from somebody or something, whether somebody sneezed on you or maybe you grabbed a door handle, that's going to be active immunity. You got exposed to the actual, infectious agent. Versus artificial, that's exogenous. That's going to be associated with vaccinations. Okay. Passive immunity, you're only dealing with those immunoglobulins, right? No B cell lymphocytes. This is with like B cell lymphocytes over here with with active. With passive, something else has already created those immunoglobulins for you. Okay. Passive natural. What's a good example of that?
Mother, like
yeah, exactly. So breast milk or IgGs. Remember IG girl that crossed the placenta? IgGs from the placenta or IgAs, right, through breast milk. That is going to be considered natural passive because you're getting those immunoglobulins straight from mom. Okay. What about active passive? Artificial passive, sorry. So passive, but artificial.
Let me see what I have on this paper for artificial passive.
Yeah, artificial passive.
So would that be like getting the rabies vaccine after being exposed to it or something?
Oh, receiving antivenom after being bitten by a rattlesnake?
Right, immunoglobulins. Anything that's immunoglobulins. A good example is the toxoid, tetanus-spasmin toxoid. What the hell is it called? Sorry, I forget it off the top of my head. But it's where you have, whoops. Oh my gosh, you guys saw my screen, didn't you?
No, I was not looking.
It doesn't matter. I recorded it.
You have to, well, you can go back and edit it, right? Like you can that part out of the video before you post it.
That gives me extra work. What's the timestamp right now? We're at, when did we start? Let me make a note of that. We're an hour in, so we're at like 55 minutes. I'll just go in there and I'll try to see if I can remove that, but that's freaking embarrassing. All right. Anyways, what's the tetanus? Tetanus. It's like a tetanus-spasmin immunoglobulin. Yeah, tetanus immune globulin. That's what it's called. And you can give that exogenously. It's called the TIG. So that is what would render the tetanus-spasmin toxin neutral. It would like neutralize it. So yeah.
So artificial passive is receiving antivenom after being bitten by a rattlesnake. That would fall under artificial.
Yeah, that would be an example of artificial passive immunity. Okay.
Got it.
Awesome. Good. Man, I can't believe I shared my screen with the exam. That's so embarrassing. All right. Anyways, moving on. Where are we here?
I don't think you did, honestly.
I was writing, so I didn't see anything. I have no clue.
I didn't see it.
You didn't see it? Okay, I'm gonna review the video and make sure that it's not in, that it didn't show up. But regardless, apparently I closed out Safari in my panic. All right, cool. We're back in. All right.
Your panic.
Yeah,
Clicking.
I panicked a little bit. Let me just make sure. That was a boomer moment. Let me make sure that I'm not sharing my screen here.
Let me see
Not
you.
sharing my screen, right? You just see me. Okay.
Yeah.
Awesome. Cool. I appreciate that. All right. So we talked about active versus passive immunity. So you should be good on that if you understood that concept that I just broke down. Let's go on through some more questions here. When we did our aseptic transfer techniques the other day, what kind of mediums did we use? We used 3 mediums. Well, what were they?
Nutrient agar, a broth.
So the nutrient agar, they're basically all nutrient agars, but there's ones that are going to be solid versus liquid, right? So you have the liquid broth right? And then those two other ones are nutrient augers as well. But one was like slanted, right? The slant auger.
Okay. Auger and then
Solid medium, right? And then the other solid medium was the actual plate, right? So if you have like a petri dish or a auger plate, nutrient auger as well, those are going to be the major types of like substrates that you can grow bacteria on. So pretty straightforward on that. Okay, and moving forward here. Hope you guys get this one right. What falls under eukaryotes? What are the different animal or organisms classes that fall under eukaryotes? What are we?
Like plant or animal?
Animalia is us. Plantae would be
Mhm.
plants. What are some of the other ones?
Fungi.
Fungi,
and-
yeah, and protozoan or protists, right?
Mhm.
So just know that those are all going to have true nuclei because that's going to be what you find in eukaryotes, a true membrane-bound nucleus. Okay. Going through them just to see which ones we should talk about. A lot of these are pretty straightforward. Oh, here's something that we didn't really talk about the other day. When we're doing inoculation or transfer of bacteria into sterile mediums, I tried to like mention this, but I even looked around and I saw that some of the students were not doing this properly. When you are like transferring bacteria onto a plate, okay, what's one thing that you want to be aware of in general?
Exposure.
Contamination. Yeah, exposure, contamination, exposure to yourself, but also you just contaminating the thing that you're trying to grow.
Yeah.
So one of the things that you want to definitely avoid doing, when you do those like different steps, when you inoculate a sterile plate, you want to very gently like remove the plate a little bit, not completely. And you definitely don't want to put the freaking lid directly on the tabletop. I saw some of the students do that the other day and I didn't say anything, but I thought it was clear when I said just open it ever so slightly so you can like put the bacteria or whatever you're trying to cultivate on there, because you could accidentally breathe on it, you could spit on it. But if you put like the lid on the table, now you got bacteria from the table and you put it back on the plate, and you're going to start growing stuff that's on the table. So you want to make sure to not cross-contaminate. The other thing that I didn't mention that I should have is that when we cultivate, when we not cultivate, but when we put the bacteria in an incubator, when we're incubating, what we do is we invert the plates over upside down. Do you guys know why we do that?
Because condensation can build.
Yeah, exactly. So when you invert it, if there's condensation, it's going to land on the lid. You're not going to have condensation landing on your bacteria, right? So that's why you turn it upside down, so you can avoid condensation, like ruining your specimen or whatever it is that you're trying to grow. All right, moving on. Moving on. We didn't talk about it yet, but okay, so someone tonight mentioned yeast infections. What's the number one cause of yeast infections?
Is it antibiotics?
Candidiasis. Yep. Candida albicans is what causes yeast infections. What kind of organism is it? Is it a virus? Is it bacteria? Is it fungus? Is it a protozoan?
Fungus?
Yeah, exactly. It's a fungus. Excellent. So we're going to go into like more fungal infections on Tuesday, but it's good for you guys to know all that. All right. Moving forward. A lot of these are pretty straightforward. We talked very briefly earlier when you mentioned active passive. I think it was active passive immunity when you have like indirect exposure or something. If you do get indirect exposure to something, say for instance you open a door, right, and you get sick from that, What's that? What's that like? What does that fall under? That's in terms of like, huh?
Like a fomite, you mean?
Yeah,
Like,
Yeah, it's like
fomite.
a fomite, and that would fall under indirect transmission, indirect contact. So make sure you know like the different types of routes of transmission. So
What is
if it's involving a fomite that you touch that's contaminated, that's going to be considered indirect contact. Direct contact is going to be shaking someone's hand, right? Coughing in someone's face. If it's like anything less than 6 feet, that's considered direct contact, right? Anything beyond 6 feet is going to be considered indirect contact. And then fomites are going to involve non-living things, whether it's an inanimate object or something that used to be alive and is now dead, like food preparation. Okay.
Really
Yes.
quick, insect is a vehicle or a vector, like a tick or a mosquito?
Good question. So there's two different things that could happen with a bug. So if a bug bites you, what do you think that would be? You have either one or two options. Is that mechanical transmission, mechanical vector, or is that going to be considered a biological vector?
Biological.
Biological.
Yeah, exactly. So if a bug bites you, a mosquito, a tick, a flea, that's going to be biological vectors, right? What if a fly lands on a turd, lands on your food, and then you eat the food?
Mechanical.
Yeah, exactly. It's going to be mechanical transmission.
Do we need to know the differences between biological and mechanical?
Totally. Absolutely. It's
Okay.
it's definitely going to be on your test.
Okay. Is that what are we talking about that on Tuesday, or did we already talk about that?
We already talked about that a little bit, but we're going to go into a little bit more detail on that like later on. But yeah, we kind of already touched those topics.
So then what would be considered a vehicle? Because isn't there fomite, a vector, and a vehicle, or is it just fomite and vector? Am I just misremembering? I could be just misremembering.
No, you're not misremembering. I'm misremembering right now off the top of my head. So vehicle transmission would be like mechanical, I think would fall under that category of vehicle. Don't quote me on that.
No, I'm just kidding.
I'd have to like go through just to verify that. I don't want to give you guys wrong information. If you understand the difference between biological versus mechanical, you should be good.
Okay.
I thought the vehicle was like a scalpel, like an infected scalpel, could be
That would
considered a
be
vehicle
an infected
transmission.
scalpel would be considered a reservoir.
But wouldn't it be considered a vehicle transmission though? I thought that's what I learned. I mean, this is like 18 years ago too.
So according to like the chains of transmission, if you had an infected scalpel blade or an infected instrument, that would be considered a reservoir. And then you would have that as having the point of entry, point of exit, and then like the point of entry. That's kind of how those chains work. It gets very, very confusing. But a scalpel blade would be considered a reservoir. Okay. And then the point of exit would be going from the scalpel blade to the point of entry, which would be like the actual laceration during the surgical procedure. In terms of vehicle, I guess the scalpel blade would also be considered a vehicle in that regard as well. But it does get a little confusing. If you understand the difference between mechanical and biological vectors, you will be pretty solid in terms of the midterm, but it does get a little bit confusing when you have to like differentiate between like the, reservoir, port of entry, port of exit, and the ultimate, host of whatever infectious agent we're dealing with.
So
I
it
just
says a
remembered that was
vehicle.
one of my board questions.
It was what?
It was like one of my board questions.
Really?
Yeah.
Which board exam did you take?
For rad tech, for X-ray.
Oh, dang, that's crazy.
So that's why I'm like, I think I remember it because I remember looking at the screen and being like, what in the actual hell is this?
Vehicle transmissions.
Yeah, so it says a microorganism transmitted to the host by contaminated items such as food, water, medications, devices, and equipment.
Right. That was under vehicle.
That's under vehicle. Yeah, so I guess anything that basically would technically be ingested or put in your skin from a mode that's not like
Right.
a
So
living object carrying.
I'm seeing here water, food, air, any of those things could be considered vehicles, which makes sense.
Yeah. Okay.
That makes sense. Sorry, I didn't know if there was a good way for me to describe that to you guys or not, but cool. All right, moving on.
Sorry. Thank you.
You're good. You're good. I'm trying to see if this one's worthwhile going through. We already kind of went over that. So let's move on. Okay, already tested you guys on that before. All right, we talked about that tonight. Sorry, I'm just like going through these. I'm just seeing which
You're
one's
fine.
good. We already talked about like staph versus strep versus like cocci versus bacilli. So I'm hoping you guys get this one correct. If you see, okay, let's just kind of recap. If you see something that's like okay, I'm going to give you the name of an organism, and you can tell me if what kind of shape it is and what kind of presentation is going to be in arrangement. Streptococcus pyogenes. What does it look like?
It looks like pearl necklace.
Yeah, exactly. A bunch of chains, a bunch of beads linked up in chains. Staphylococcus aureus.
Clusters of grapes.
Good. Excellent. E.
Oh.
coli.
Bacillus.
Yeah, good. Gram-negative bacillus. What about lactobacillus?
Is that the chains of bacillus?
Yeah, it's going to be mostly like a streptobacillus, right? It's going to be streptobacilli all in chains. If you see Streptococcus pneumoniae, even though it's called a streptococcus, actually, here's a better example because this is always going to be in this shape. Neisseria gonorrhoeae. What does that look like?
Cocci.
It is cocci. And how do they arrange themselves?
Are they diplo?
Yeah, that's awesome. Good job. They're going to hang out in twos. Neisseria gonorrhoeae, as well as Neisseria meningitis, are going to hang out in twos. The way that I remember Neisseria gonorrhoeae, it's going to be gonorrhea, gonads, twos. Okay. That's the way I remember Neisseria gonorrhea. Yeah, exactly.
Okay.
I know it's like a really rude way to remember it, but yeah, it works.
I mean, none of us are gonna probably get that wrong.
That's a good way to remember it. Okay, anyways, moving on. That was awkward. Let's see. We talked about the iodine and Gram staining. What's another term for that?
Morton.
Yeah, good job. All right. So there's going to be different types of toxins and different types of bacteria. So let's go through like different virulence factors that are going to be associated with a bacteria. What is the term for a toxin that's going to be secreted by the bacteria? A good example would be like exfoliatin, toxic shock syndrome. There's like a toxin associated with that as well. Hyaluronidase. These are things that are secreted. What are those called?
Exotoxin.
Yeah, exactly. If they're secreted, it's going to be called an exotoxin. If it's associated with the bacterial structure, think like the lipopolysaccharides, lipid A, O antigen. What are those called?
Endotoxins.
Good. Excellent. Now, what about other virulence factors? Give me two more virulence factors. We talked about them already tonight. We talked about them just the other day.
Actually just wrote about one too. Is it like replication or growth?
Surrounds the bacteria.
Oh, the The capsules.
And then also another virulence factor would be considered biofilm too, which is kind of like similar to capsules in terms of structure. So good. I'm glad you guys know the difference between all those. That's awesome. Here's one that we have not yet really gone into great detail about, but I just talked about just recently. But okay, Streptococcus pyogenes. You said pearl necklace. What kind of pathologies does strep pyogenes cause? Causes a lot of things, but what's like the main thing that strep pyogenes causes? I've mentioned it in class and lecture in passing. We're going to go into more detail about it later on, but if you remember this, I'll give you a huge high five.
Like, what does it cause within the body?
Yeah, I mean, think pearl necklace, strep pyogenes.
Strep throat.
Inflammation.
Yeah, strep throat. Yeah.
Oh, I didn't know what you were asking. I was like,
Yeah.
what? Like,
Yeah, that's why I'm like,
inflammation in your throat, strep throat, redness in the throat, like.
Yeah, strep pyogenes is like the number one cause of strep throat, but it also causes a lot of other things. It can cause impetigo, which are going to be like these honey crust little lesions around the mouth. Happens a lot in children. It can cause toxic shock syndrome. It can cause necrotizing fasciitis. It can also cause scarlet fever, which can eventually turn into rheumatic fever, which involves the heart. So it causes lots of different things. We're going to talk about strep pyogenes later on. But if you know that strep pyogenes is the number one cause of strep throat, you'll be good for now. We're going to talk about strep pyogenes later as well, but just know that for now. What are the two major categories of antibiotics? I don't know if we really talked about this in class yet, but we're going to talk about it in chapter 5. There are ones that inhibit growth, and there are ones that kill the bacteria.
Oh, bactericidal and bacteriostatic.
Yes, the ones that kill bacteria are bactericidal. The ones that inhibit growth are going to be bacteriostatic. Bactericidal, great example, beta-lactams, penicillins, cephalosporins. Those are going to be bactericidal. They kill the bacteria. Anything that's going to irreversibly bind to the bacteria is going to be bacteriostatic. A really common class of drug, well, not class of drug, but your sulfa drugs, so sulfamethoxazole, trimethoprim, those are bacteriostatic. So that combination, trimethoprim, sulfamethoxazole, is called Bactrim. You'll see that prescribed a lot in clinic, okay? It's very commonly used, especially for like UTIs. It prevents the bacteria from growing. What happens though is that your body just flushes the bacteria out mechanically, right? So through urination, or the body through immunological response is able to like phagocytose and get rid of the bacteria, eliminate it through natural means. So that's the difference between bacteriostatic versus bactericidal. Okay, we already went over that. We went over that the other day.
How many questions are on this midterm?
75 questions.
The huge.
Yep, they're both going to be 75 questions. Okay, moving on. These are all pretty straightforward. We already covered all these bases. Okay. Okay, so what's the term for, let's talk about like a viral infection. What's the term for the first onset of symptoms of a viral infection? What phase is that? You're sick and then you recover. What's that called?
Did you freeze, or are you staring at us now?
I'm waiting for you guys to answer the question.
Are you talking about like when there's like the period where you get a temperature, and then the period where the signs and symptoms express themselves, and then there's like the highest?
Yeah.
Well, I do know this.
So when it first starts, when it first happens, what's that called? Do you like get sick? You recover? Starts with an A. It's a short period of time.
Acute?
Yeah, exactly. That's an acute infection. What happens if it does that? It goes away, but then it comes back again. What's that going away period? That in between? Huh?
Latency.
Latency. Awesome. What's it called when it comes back again?
Chronic.
Sort of. There's a term specifically for that part when it comes back again.
Tell us what letter it starts with.
It starts with an R.
Reoccurring.
Yeah, reoccurring or reactivation. Reactivation.
Okay,
So
so
herpes
acute.
is a great example. You have the acute latency gets reactivated, right? Then what happens? You already said the other C word, right? So you get sick, and then eventually you start seeing persistent symptoms, right? So that's going to be
Chronic.
chronic. Exactly. Awesome. Good job on that. Let's see. What are two pretty like common protozoan type infections that you get from drinking contaminated water? Causes diarrhea, one of which causes bloody diarrhea.
Montezuma's Revenge.
Well, Montezuma's Revenge is actually E. coli. So it's a little bit different. That's a bacterial infection. The proper terminology would be beaver fever for the first one, which is my favorite term for a disease, beaver fever. It's kind of hilarious. You get it from drinking contaminated water when you're on a hike. Dogs get it. Animals get it. We talked about it the other day.
Giardia.
Yeah, Giardia. Giardia is one. What's the other one? The other one causes like really aggressive bloody diarrhea. It's another protozoan. It causes bloody diarrhea. It destroys tissues in your GI tract. Okay. Okay. So entamoeba histolytica. So entamoeba, right? Enteric amoeba, right? Inside your GI. Histolytica. Histo, tissues, lytic, destruction of. Okay. So GI amoeba that destroys tissues, causing bloody diarrhea. Okay, so Giardia, entamoeba histolytica. Those are like two severe GI type protozoans that are relatively common.
Yeah, that Giardia cost me like $3,000 with my dog.
Oh my God. Is that how much the metronidazole cost?
She couldn't get rid of it. It took almost a year to get rid of.
Despite
So
taking metronidazole?
Yeah, she's on. So she went in 4 times.
And
Because
metronidazole is a cheap drug. How is it that it costs that much? Was it the actual vet visit? Because that would make
Yeah, everything.
Yeah, because the drug itself is pretty freaking cheap. Metronidazole is also a very cheap drug.
Yep, it was like the testing of it and everything else. Yeah, she's my
5,000.
rescue $10,000 dog.
Woo! Ouch. That hurts. That really hurts to hear. Yeah. I want to get a dog again, but that's one thing I don't want to deal with is all the costs. All right, we haven't talked about helminths yet, but what's a helminth?
Worms.
A worm. Excellent. Let's go over some of the different types of worms. What is a worm that you would see hanging out maybe near the duodenum, around the liver? It consumes bile, maybe.
Tapeworm.
Tapeworm.
No, not yet.
Hookworm.
Flukes. Yeah, awesome. Fluke worm is a good example of that. So flukes are going to be hanging out. They're usually like consuming like bile. They're called liver flukes. Tapeworm. Now that you already mentioned it, there's 2 major types of tapeworms that we deal with. One is Tinea saginatum. One is Tinea solium, right? One is beef. The other one's pork. There's other tapeworms too, like Diphyllobathrum latum. Don't worry about the names. What about worms that cause an itchy perianal itching? I'll be technical about the terminology. Or itchy butt. What's the term for that one? Do you guys know?
Let's start with a P.
Yeah. Pin the tail on the donkey. Pinworm.
Pinworm. Yeah.
My previous students of mine came up with that mnemonic, and I love it. Pin the tail on the donkey. It causes perianal itching because in the middle of the night, the worms sneak out.
No.
They
Stop.
lay their eggs around.
Absolutely not.
And that's what causes the itching. It happens a lot in children. You guys know how to diagnose it?
No. Please
Yeah.
tell me, because if one of my children has this itchy butthole with
Scotch tape test. You literally put a piece of Scotch tape around the anus. You bring the Scotch tape back to your doctor the next day. They look at it underneath the microscope. If you see eggs or if you see worms, then you know that the patient has pinworm, and then you can give them a drug. Quite frankly, you could probably just treat them with an antihelminth like albendazole or something, just knowing what the symptoms are.
I can't think of a kid on earth that will fall asleep with Scotch tape on their butthole.
That's hilarious.
I know my kids would not be having any of that.
Yeah, but to deal with the actual itchiness itself, I'm pretty sure that they would probably deal with it just if it helps, you know.
Try doing a BE on children where you can't pump up the balloon to inflate it to stay in. You have to tape it all down. Yeah, that's real fun. I was covered head to toe countless times in fecal material. Yeah.
Oh, that's absolutely awful.
This is why I don't have kids. See?
And this is why you don't want to go into clinical practice, right? Don't you want to go a different route?
I have stories. I could write a book.
Yeah, no, that's awesome. Do it. Do it and let me know when it's published so I can
maggot therapy. Oh.
No, they used to do that in the ICU when I worked at Banner. They do the maggot and the leech treatment on the patients.
I love it. I love it. Medicine is fabulous. It's a lot.
What about ringworm? Is that very
ringworm is actually not a worm.
Okay.
It's a fungus.
It's a fungus.
Ringworm is a fungus, but it looks like a ring and it kind of looks like a worm. So the name stuck. But the other one is hookworm. And hookworm, when we get into the helminths, I usually put a picture of a child walking in grass barefoot. It's going to be associated with stepping on a turd barefoot. And so the hookworm literally penetrates through your skin, gets into your circulatory system, and eventually migrates its way into your GI tract.
I saw the foot of this kid that they went down to like Florida on vacation or something, and they, just buried him in the sand. You know how you do what the kids do at the beach? Getting hookworm on his foot, and like the progression of it was so gnarly that I don't even want to take my kids to the beach anymore.
I'm going to the beach?
Yeah, at the beach, because people will walk their dogs and run their dogs on the beach,
that's awful. I would have never thought. I would have never thought walking the beach would predispose you to hookworm, but that's the thing about microbiology and medicine in general. It will take the fun out of the simple things in life.
That is what I'm saying. Like, I'm convinced anytime, like, whatever, my toe itches, I have hookworm in my foot now, or because I walked outside with no shoes on after. Like,
you probably have hookworm for sure.
I probably do. I also have a tapeworm. I'm gonna feel like I have pinworms tonight when I go to bed.
I think you also have hypochondriasis. So.
Yeah, 100%. Yeah, only after taking these classes, though.
Funny.
I'm totally disturbed now.
All right, I know. Yeah, it's a disturbing field, right? Medicine is very disturbing. You learn about all the things that will kill you. It turns out that nature wants to just chew you up and spit you out. All right. Something that we don't really talk about in this class is Rocky Mountain spotted fever. We also don't talk about some of the other biological type vectors, for instance, like ticks. We do talk about plasmodium and things like that, but no, I'm just going to give this to you guys. If you hear Rocky Mountain spotted fever, that's going to be an infection transmitted by a tick bite. And if you get bit by a bug and you get an infection from that, what's that called? What kind of vector?
Biological.
Yeah, it's a biological vector. Exactly. All right. Moving forward. We're
Sounds
in
like we
final
got a lot.
stretch here. We're in the final stretch. I'm sorry, what'd you say?
It sounds like we got a lot of gimmies on this.
We got a lot covered because there's a lot of crossover. There'll be like one question that talks about a certain topic, and there'll be another question about the same topic but restructured. Okay, so you get like a lot of like rinse and repeat in terms of like content. Damn, we covered a lot of this stuff. We covered that too. Wow, we've really covered a lot of ground so far.
So basically, don't get thrown off if there are similar questions that have
Yeah,
the same answers that are just
exactly. Yeah,
Okay.
exactly. Try not to get thrown off because I see the percentiles of the people who answer the questions incorrectly, and some of these are pretty high for incorrect answers, even though they're simple. But I'm trying to like go over the ones that I think are going to be like more high yield for you guys. When we did our little like pairing up, like share and pair the other day, some of you guys did fungi, okay, and some of you guys did fungi that affected your toenails. Someone else talked about athlete's foot. Okay, so well, I guess I kind of already gave that away. What is a term for those types of fungus that affects your skin? What's your skin referred to as? Starts with a D. Derma. So if it's something affecting your skin and it's like a fungal type infection, it's a dermatophyte. Right? Dermatophytes. You guys heard of that term before, right? So there's a lot of different dermatophytes. There's dermatophytes that are going to affect your skin, right? That cause tinea cruris, which is like jock itch, tinea pedis, which is your feet, right? Athlete's foot. Tinea capitis that affects your like hair follicles, your scalp. There's onychomycosis, which affects your nails, kind of falls under a similar branch. So those are some major types of like tinea type infections, but they're all going to be fungal in nature. So when you see something that's like a dermatophyte, you're thinking a fungal type of infection. Okay. We already went over this. What's another term for valley fever?
Jen.
Coccidioidomycosis.
Yeah, coccidioidomycosis, coccidioides immitis. It's also referred to as San Joaquin Valley fever. Okay, we talked about that.
What is it? Coccidio what?
Coccidioidomycosis.
That was the only question I got wrong last night.
Oh, good for you.
I put the bacillus one because I don't know why.
That's funny.
Okay.
That's funny. We're at the final stretch. 5 more here. Final stretch. You guys better get that one correct. That's pretty straightforward. If an organism is an obligate anaerobe, what does that mean?
It needs air, oxygen.
Oxygen. If it's an obligate aerobe, what does that mean?
That it can survive without oxygen.
No, it's an obligate aerobe. It's obligatory.
Right.
It requires oxygen.
Requires oxygen.
If it's like a facultated anaerobe, that means that it can exist without oxygen, but it can also exist without oxygen. If it's a microaerophile, it's going to be something that requires just a little bit of oxygen. But if you know aerobe versus anaerobe, you'll be solid when it comes to oxygen requirements. Okay, that's pretty straightforward. 2 more. Oh, this one's kind of weird. That's a really weird question. This one's very strange, but I'm gonna try to restructure this and explain it so that you guys, when you see the question, you'll kind of understand where you're coming from here. so you remember we talked about barriers of entry, right? As being part of that like innate immune system, right? So those are going to be what? Skin. What else? Membrane. So that could be like inside your mouth, your GI tract, could be inside your intestines, could be also genitourinary tract, right? So if you have like a bacterial infection that's causing a UTI, there's going to be a little bit of a protection from your barriers of entry, right? So the mucosal membrane of your genital urinary tract. So just keep that in mind when you think about UTIs. If you also think about the way that you eliminate those organisms when you take a bacteriostatic compound like Bactrim, whatever, you're going to be depending a lot on that barrier of entry. Okay. So if you see that question, just keep that in mind. Okay. Without me giving that question away. What happens with binary fission? What is binary fission? What does that mean?
How bacteria replicate.
Yeah.
Yes. What is the consequence of binary fission? You start with one parent cell, and then you have what?
2 daughter cells.
And what are they? Are they different? Are they the same?
Identical.
Yeah, they're supposed to be identical. You will see very... That's so cute. Okay.
What is it?
The dog.
Let me see.
Doggo. So you do see sometimes mutations taking place, right? And that's why you see like development of like antibiotic resistance, things like that, right? Because binary fission happens so rapidly. It's exponential, logarithmic. The doubling time is cut in half every single time you have reproduction. And so because of that really rapid rate of nucleotide base pairing, you're going to get a lot of random mutations as well. But theoretically speaking, both of those daughter cells are supposed to be genetically identical. So that's another key component of binary fission. That's basically it. That basically summarizes all of your midterm. So with that being said, I scheduled this for 2 hours. We're at 6:30. We're at an hour and a half right now. Is there anything else that you guys want further clarification on? Okay. So I'm going to post this video tonight on YouTube. I'm going to upload. I'm going to have it like unclassified. So your images and stuff, no one's going to see it. Only people with the link, AKA you guys in Canvas. So I'm going to post this to the rest of the class. You can re-listen to it as you wish. And if you have any other questions, just shoot me an email. Emailing is the best way if it's like, if you really want me to get back to you. Otherwise, just shoot me a message in Halo. Sorry, not Halo. Sorry, that's what GCU uses. Shoot me a message in Canvas. So if you don't have any other questions,
No, I think I'm good.
yeah, if you're good, then you're dismissed. Have a wonderful night. And
You too.
Thank you so much.
Actually,
Yeah.
can you post the quizzes questions from yesterday?
Yes.
did we finish them? Did we go through every
Yeah.
single one?
Yeah, we got
Yeah.
21.
Yeah, if we finish
Yeah.
them, I'll go ahead and post them. I'll have them
Cool.
all posted for you tonight. I'll post a link.
Awesome.
Love the
Thank
video.
you.
Yeah, absolutely. My
Check
pleasure.
your video at 5:55.
Sorry?
So don't forget to check that leaked exam at 5:55.
Ah, yes. I'm going to check that right now. Thanks for the reminder.
All right. Thank you so much.
All right, y'all. Have a great
Good
night. Take
night.
care.
Good night.
Cheers. All right. Bye-bye.